For most of the history of human genetics, Pakistan barely existed on the map. Not the physical map, the genetic one. While researchers spent decades building enormous genetic databases from European and East Asian populations, cataloging millions of genomes, and using them to power everything from disease risk calculators to new drug development, Pakistan's more than 240 million people remained almost invisible in that global scientific record, a genuine blind spot sitting at the heart of one of the most genetically diverse regions on Earth. This absence was never simply an oversight or a matter of low priority. Building a large-scale national genome database requires enormous infrastructure, sequencing facilities, trained researchers, secure data systems, sustained funding, and years of coordinated institutional effort. For most of the 20th century and well into the 21st, that kind of infrastructure existed almost exclusively in wealthy nations with long-established biomedical research traditions, leaving countries across South Asia, the Middle East, and much of Africa chronically underrepresented in the genetic data underpinning modern medicine, despite collectively representing a majority of the world's population. Then scientists decided to fix that gap, and what they built along the way became something far bigger and far stranger than anyone expected. Researchers assembled and analyzed more than 170,000 Pakistani genomes, by a wide margin, the largest genetic study ever conducted on any South Asian population, and one of the largest single-country genomic datasets ever assembled anywhere in the world. When they compared this enormous dataset against global genetic databases built primarily from European, East Asian, and other well-studied populations, they didn't just fill in a missing piece of the world's genetic map. They found something that set Pakistan apart from virtually every other country ever studied at this scale. A population carrying the largest documented collection of fully inactivated human genes found anywhere on Earth. This is the story of what scientists found when they finally compared 170,000 Pakistani genomes to the rest of the world. To understand why this finding matters so profoundly, you first need to understand a basic and slightly strange fact about human genetics that most people never think about. Every person carries 2 copies of nearly every gene in their body, one inherited from each parent. In the vast majority of cases, if one copy of a gene happens to carry a harmful mutation, the second, healthy copy compensates, and the gene still functions normally. Geneticists call this recessive protection, a kind of built-in backup system embedded into human biology. But when 2 people who are closely related and therefore likely to carry the same rare harmful mutations inherited from a shared recent ancestor have children together, that backup system starts to fail. If both parents happen to carry a mutation in the same gene, there's a meaningful chance their child will inherit 2 broken copies instead of 1 broken and 1 healthy, completely silencing that gene's function. Scientists call this a genetic knockout. A gene switched off entirely, its protein coding instructions no longer producing anything useful at all. This is precisely where Pakistan's genetic story becomes scientifically extraordinary, and it connects directly to a demographic reality documented for decades in the country. Exceptionally high rates of consanguineous marriage, meaning marriage between close blood relatives, most commonly first cousins. Depending on the specific survey and region examined, estimates of consanguineous marriage in Pakistan have ranged from roughly 40 to 60% of all marriages nationwide, among the highest rates recorded anywhere in the world, reflecting deeply rooted cultural, social, and economic traditions that have shaped family and marriage patterns across the country for generations, often tied to preserving family property, strengthening kinship bonds, and maintaining social and economic stability within extended family networks. This is precisely where Pakistan's genetic story becomes scientifically extraordinary, and it connects directly to a demographic reality documented for decades in the country. Exceptionally high rates of consanguineous marriage, meaning marriage between close blood relatives, most commonly first cousins. Depending on the specific survey and region examined, estimates of consanguineous marriage in Pakistan have ranged from roughly 40 to 60% of all marriages nationwide, among the highest rates recorded anywhere in the world, reflecting deeply rooted cultural, social, and economic traditions that have shaped family and marriage patterns across the country for generations, often tied to preserving family property, strengthening kinship bonds, and maintaining social and economic stability within extended family networks. To put that percentage in global context, most Western countries report consanguineous marriage rates well under 1% of the population, meaning the difference between Pakistan and many comparison populations used to build existing global genetic databases isn't simply a matter of degree. It represents an entirely different demographic regime, one that produces a measurably different genetic landscape. This is exactly why researchers building the world's major genetic reference databases over the past several decades, drawing overwhelmingly from populations with very low consanguinity rates, ended up with datasets containing remarkably few naturally occurring examples of complete gene knockouts. It isn't that those populations lack genetic mutations. Every human population carries plenty of them. It's that the specific demographic conditions required to reliably produce double recessive gene knockouts, generation after generation, at a scale large enough for rigorous scientific study, happened to be dramatically more common in Pakistan than in the populations that historically dominated global genomic research. When researchers built the Pakistan Genome Resource, a national biobank specifically designed to capture and analyze this genetic reality at scale, they found the consequences of this demographic pattern written directly and unmistakably into the DNA of the population. Across the more than 170,000 genomes analyzed, researchers identified, on average, one complete loss-of-function gene knockout for every 4 individuals sequenced, an extraordinarily high rate compared to genetic databases built from populations with lower rates of consanguineous marriage, where complete gene knockouts of this kind are considerably rarer events, typically appearing at a fraction of that frequency. The scale of this finding, once you sit with it, is genuinely remarkable. It means that across the full Pakistan Genome Resource dataset, researchers cataloged what they describe as the largest pool of complete loss-of-function mutations ever assembled from a single population's genomic data anywhere in the world. Tens of thousands of individuals, each one a natural, real-world example of what happens when a specific human gene simply stops working entirely, information that medical researchers elsewhere have historically had to try to approximate using laboratory-engineered mice, genetically modified specifically to knock out one gene at a time in painstaking, expensive, and often imperfect experiments that don't always translate reliably to actual human biology. This is precisely why researchers studying human genetics worldwide have started paying very close attention to exactly this kind of data set. Naturally occurring human gene knockouts found in real people living real lives rather than created in a laboratory offer scientists something genuinely rare and valuable: direct evidence of what a specific gene actually does in a living human being when it stops functioning altogether. For decades, understanding gene function in humans has depended heavily on animal models, particularly mice, because deliberately disabling a gene in a living human being for research purposes is, obviously, never ethically permissible. Populations with elevated rates of consanguineous marriage effectively provide researchers with a naturally occurring version of exactly this kind of experiment, conducted not in a lab, but through the ordinary course of family life across many generations, offering real, ethically sourced human data about gene function that researchers elsewhere simply cannot obtain any other way. The research, led by scientists including Columbia University's Danish Salehin, working in close collaboration with institutions across Pakistan, didn't stop at simply cataloging these genetic knockouts. The entire point of building a resource like this is to connect specific, inactivated genes to specific, measurable effects on human health. Information with direct, practical implications for understanding disease risk, drug safety, and the development of new medical treatments. When a specific gene knockout appears at meaningfully elevated frequency within a well-documented population, and researchers can then study the health outcomes of the specific individuals carrying that knockout, they gain a uniquely powerful natural experiment for understanding precisely what that gene does in real human physiology. Information that pharmaceutical companies and medical researchers around the world have started actively seeking out, precisely because it's so difficult to obtain through any other research method. This kind of research carries a particular, almost paradoxical significance once you consider it in full, a demographic and cultural pattern sometimes discussed primarily in terms of its health risks, because consanguineous marriage does measurably increase the likelihood of certain recessive genetic disorders appearing in a population, has, through large-scale genomic research, also become a genuinely significant scientific asset, offering the entire world insight into human gene function that few, if any, other populations could provide at this scale. Pakistan's genetic story, in other words, turns out to be considerably more scientifically valuable to global medicine than its historical absence from genetic databases ever suggested it would be. The Pakistan Genome Resource study didn't emerge in isolation either. It sits alongside a broader wave of major South Asian genomic research efforts that have collectively transformed the region's presence in global genetic science within just the past 2 years. Earlier in 2026, India's own National Genome India project completed sequencing of nearly 10,000 individuals from across the country, uncovering roughly 130 million genetic variants, close to a third of which had never before been documented in any global genetic database, alongside tens of millions of additional variants entirely absent from the Eurocentric genomic databases that have historically dominated medical research. Around the same period, a separate Indian Genetic Atlas project, funded through the country's Department of Biotechnology and coordinated across 20 research institutions, built its own detailed map capturing rare genetic variants specific to individual regional and community populations across India, reflecting the same long history of migration, isolation, and endogamous marriage that shapes genetic diversity across the broader subcontinent. Taken together, these efforts represent something genuinely historic for South Asian genetics as a field. A coordinated, large-scale correction to one of the most consequential gaps in modern medical science. South Asians collectively make up nearly 25% of the entire world's population, yet for decades represented barely 2% of the genetic data contained in the world's major genomic databases. A mismatch researchers have repeatedly identified as having direct, measurable consequences for how well modern medicine actually serves well over 2 billion people. Genetic reference databases built almost entirely from European populations have for years produced disease risk calculations, drug dosage guidelines, and diagnostic tools that simply don't translate accurately to South Asian genetic backgrounds, sometimes leading to misdiagnosis, ineffective treatment, or misidentification of genuine health risks specific to populations whose genetic variation those databases were never designed to capture in the first place. It's worth pausing to appreciate the sheer scale of what assembling 170,000 genomes actually required, because a project of this size represents a monumental logistical and scientific undertaking, not simply a single dramatic discovery. Building the Pakistan Genome Resource meant recruiting participants across the country, obtaining informed consent, collecting samples, sequencing each individual genome, and then running the computational analysis needed to identify and confirm genetic knockouts across a dataset. This enormous, a process requiring years of sustained institutional collaboration, significant funding, and the kind of large-scale genomic infrastructure that, until relatively recently, essentially didn't exist anywhere in South Asia at this scale. The fact that Pakistan now possesses one of the largest single-population genomic datasets in the entire world represents a genuine scientific achievement, transforming the country almost overnight from one of the more genetically understudied major populations on Earth into a global reference point for an entire specific category of medical genetic research. The research leadership behind this effort reflects exactly the kind of sustained international collaboration such a large undertaking demands. Danish Salehin, a Pakistani-born epidemiologist and geneticist now based at Columbia University in New York, has spent much of his career specifically focused on building genetic research infrastructure, connecting Pakistani institutions to the broader global scientific community. Recognizing early on that Pakistan's specific demographic profile, including its notably high rates of consanguineous marriage, represented an underexploited scientific opportunity rather than simply a public health statistic. That long-term vision, sustained across years of institutional relationship building, grant funding, and technical infrastructure development, is precisely what made a dataset of this unprecedented scale possible, transforming what began as domestically focused public health research into a resource now drawing serious attention from the global pharmaceutical and genetics research community. A concrete historical example helps illustrate exactly why pharmaceutical researchers care so intensely about this kind of data. One of the most successful new classes of cholesterol-lowering drugs developed over the past 2 decades, a category known as PCSK9 inhibitors, traces its scientific origin story directly back to the discovery of a small number of individuals who naturally carried loss-of-function mutations in the PCSK9 gene, and who, researchers found, had dramatically lower lifetime cholesterol levels and correspondingly lower rates of heart disease as a direct result. That real-world human evidence gave pharmaceutical companies the confidence to invest heavily in developing drugs designed to artificially replicate that same effect in patients who didn't naturally carry the beneficial mutation, drugs that have since helped prevent heart attacks and strokes in patients worldwide. The entire drug category exists, in a very real sense, because scientists were able to study real people whose bodies had already run the experiment naturally. A dataset the size and depth of the Pakistan Genome Resource, containing tens of thousands of similarly naturally occurring gene knockouts across a huge range of different genes, offers researchers the raw material to potentially discover dozens, if not hundreds, of similar drug development opportunities, each one starting from the same basic principle. Find the people whose bodies have already shown scientists what happens when a specific gene goes silent. There's a broader significance to this finding that extends well beyond Pakistan's own borders, touching on how modern medicine develops new treatments for the entire world. Pharmaceutical researchers have increasingly recognized that studying naturally occurring human gene knockouts offers one of the most reliable ways to predict what happens when a drug is designed to deliberately target and inhibit a specific gene's function. Because a population that already contains real people missing that gene entirely provides a direct, observable answer to the question every drug developer needs answered before investing years and billions of dollars into a new treatment. What actually happens to a person's health when this particular gene stops working? Some of the most successful drugs developed in recent decades, across categories ranging from cholesterol management to pain treatment, trace their scientific validation directly back to genetic studies of populations with naturally occurring loss-of-function mutations in the genes those drugs were ultimately designed to target. Pakistan's uniquely large collection of documented gene knockouts positions the country's genomic data as a genuinely significant resource for exactly this kind of pharmaceutical research going forward, with direct relevance not just to treating diseases specific to Pakistan's own population, but to drug development efforts serving patients worldwide. It's worth approaching this topic with genuine care, because consanguineous marriage carries deep cultural and social significance across Pakistan and much of the broader Muslim world, rather than representing some kind of aberration or oversight. Marriage between cousins has been practiced across many cultures throughout human history, and remains a deliberate, valued tradition in Pakistan today, often serving important social functions, keeping family wealth and property consolidated within extended kinship networks, strengthening bonds between related families, ensuring compatible social and economic backgrounds between spouses, and providing a sense of security and familiarity that many families consider genuinely important when arranging marriages for their children. Researchers studying this topic are careful to note that documenting the genetic consequences of this practice is not the same as passing judgment on the cultural tradition itself. It is simply an accurate scientific description of one specific, measurable biological outcome connected to a demographic pattern that exists for entirely separate cultural, social, and economic reasons. None of this minimizes the very real public health challenges that elevated consanguineous marriage rates present within Pakistan itself. Researchers studying this pattern have consistently documented higher rates of certain recessive genetic disorders in populations with high rates of cousin marriage, findings that have informed ongoing public health conversations within Pakistan about genetic counseling, prenatal screening, and family planning resources, particularly in communities where consanguineous marriage remains a deeply established cultural tradition. The Pakistan Genome Resource itself, beyond its broader scientific value to global research, was explicitly designed with this domestic health application in mind, giving Pakistani researchers and healthcare providers a far more detailed, population-specific genetic reference than has ever existed before, better equipping the country's own medical system to identify, understand, and eventually help address the specific genetic health risks its population actually faces, rather than relying on diagnostic tools calibrated for entirely different populations elsewhere in the world. What makes this story genuinely striking when you step back and look at the full picture is how a demographic pattern often discussed in narrow terms, as either a cultural tradition or a health risk factor, depending on who's doing the discussing, turns out, once examined through the lens of large-scale modern genomics, to be neither simply one or the other, but something considerably more complex. A genuine, measurable contribution to global medical science, generated through Pakistan's specific demographic history, now positioned to help researchers worldwide better understand human biology in ways that populations with lower rates of consanguineous marriage simply cannot replicate at the same scale or clarity. There's a broader pattern worth recognizing here, one that connects this Pakistani genomic breakthrough to a wider story unfolding across South Asia and much of the developing world. For most of the 20th century, the flow of genetic and medical knowledge ran overwhelmingly in one direction. Wealthy nations with established research infrastructure generated the data, developed the drugs, and published the studies, while the rest of the world largely served as a market for treatments developed using genetic information that didn't necessarily reflect their own population's biology. The Pakistan Genome Resource, alongside similar large-scale efforts now underway across India, Bangladesh, and other South Asian nations, represents a genuine shift in that historical pattern. Countries once treated primarily as recipients of Western medical research, increasingly positioning themselves as genuine contributors to the global scientific knowledge base, generating data and insights that researchers in wealthy nations now actively seek out, rather than simply importing. This shift carries real symbolic as well as practical weight. For decades, global health and genetics research operated on an implicit assumption that scientifically valuable data came primarily from certain parts of the world, while other regions existed mainly to receive the resulting medical treatments. A dataset like the Pakistan Genome Resource complicates that assumption directly, demonstrating that a country long treated as scientifically peripheral to global genetics research actually possesses one of the single most clinically valuable genomic datasets currently available anywhere on Earth, specifically because of a demographic characteristic unique to its own population history. That reversal, from data recipient to data originator, reflects a broader transformation reshaping global genomics research, one study, one national genome project, and one previously overlooked population at a time. For scientists, that is the result that emerged when they finally compared 170,000 Pakistani genomes against the world's existing genetic databases. Not simply confirmation that Pakistan, like every nation, carries its own distinct genetic story worth documenting, Researchers expected that much going in. What genuinely surprised the broader scientific community was the sheer scale and clinical value of what that comparison revealed. The largest documented collection of naturally occurring human gene knockouts ever assembled from a single population. A genuinely unique scientific resource born directly from Pakistan's specific demographic history, now poised to help researchers around the world better understand human gene function, develop safer and more effective drugs, and finally begin correcting a genetic data gap that has left over 1 billion South Asians underserved by modern medicine for far too long. That is what scientists found when they finally compared Pakistani DNA to the world. Not simply another entry in the growing catalog of national genome projects, but a population whose unique genetic history, shaped by centuries of specific marriage and community patterns, turned out to hold scientific value far beyond its own borders, offering the entire world a window into human biology that few, if any, other populations on Earth could have provided, and a genuine reminder that the most valuable scientific resources sometimes come from exactly the places the world spent decades overlooking.